Welcome, everyone, and thank you all for being here. On behalf of Women Abroad and Equity in Biomedicine, we're thrilled to have Dr. Nyasha Chambwe here with us today to talk about racial disparities in cancer research. Nyasha Chambwe, PhD, is an assistant professor at the Institute for Molecular Medicine at the Fireside Institute for Medical Research. In 2014, she received her PhD in computational biology from Weill Cornell Medicine in New York. She then went on to complete her postdoctoral training at the Institute for Systems Biology in 2019 with the late Dr. Alicia Martin in cancer integrative genomic analysis and biomarker discovery. Dr. Chambwe analyzes patient-derived datasets in order to understand the risk for cancer development and progression, how tumors respond to treatment, and how to better predict effective treatment regimens to improve cancer outcomes. A particular focus of her work is in understanding the biological basis for cancer health disparities experienced by understudied populations, particularly those of African descent, and with an eye toward informing better interventions that will lead to equitable health outcomes. Thank you all for being here.
Thank you for that wonderful introduction, Ro. Good afternoon to everybody. This afternoon, we're going to have a discussion, a provocative discussion about a topic that is close to my heart and to the heart of many people. And we've set up the session this afternoon. I'm going to give a short overview talk of this area of cancer disparities. So it's a quick overview talk, just introducing concepts and the general motivating the problem. And then Dr. Martin and I are going to have a chat, a fireside chat. It's rather cool here in Boston this afternoon. We have the chairs, but not the fire. But hopefully we'll bring the fire with the discussion. And of course, we'll at that point open it up for discussion to the room. Okay, and so to get started, I thought we'd just start with a definition, okay? Cancer health disparities are differences in disease measures between population groups. So you can consider the measures as incidence or prevalence or mortality or morbidity, et cetera, as listed on the slide. And we typically, when you hear cancer disparities, it's easy to jump immediately to racial and ethnic disparities. But I want to highlight at this juncture that there are many population groups who experience outcomes that are not positive compared to other groups that are defined on many axes of how they self-identify. So that includes by socioeconomic status or by their individuals with disability or how individuals identify by their sexual or gender identity. So I don't want us to leave this room this afternoon believing only that people of racial and ethnic minorities in the US are the only ones who experience disparities. There are inequities in many population groups across the board in cancer, and so I want us to keep that in mind. My group studies racial and ethnic disparities, and these are well documented and have persisted for a long time. So this is data from the NCI SEER program, which epidemiological surveillance database, where we're looking here at mortality, age-adjusted mortality from 2000 to 2019 by race and ethnicity, so how people self-identify. And we can see that although in general, mortality due to cancer across different sites is going down, that there are still persistent differences. If we look here at the end of the graph here, when this last data was collected in 2019, that mortality is higher in certain groups that identify, for example, here, non-Hispanic groups have the highest mortality in cancer across the board in the US. Just so that we are not singularly focused on the US, we also see just abundant global variation in cancer incidence and mortality globally. This data is looking at the annualized rate of change in terms of cancer incidence over the same sort of 10 years as we looked at in the previous data. And we're seeing a recent trend where we're seeing in low and middle income countries a rapid increase in cancer incidence that is projected to be very, very high over the next 30 to 50 years, according to UN WHO data. So this problem of differences in cancer outcomes and incidence is predicted to be an even major public health challenge worldwide. If we consider the income levels of different countries across the globe, we see that the estimated new cancer cases, those rates are different according to whether or not the countries are classified as high HDI, so sort of their socioeconomic development index, so high HDI countries versus low HDI countries. Basically, poorer countries are expected to have a much larger incidence are projected to have much larger incidence of cancer. So if you consider some of these factors driving disparities across groups, whether it's by socioeconomic status, how people self-identify by race or ethnicity, where people live, etc., the issue of cancer health disparities is not germane or unique to the US, but is projected to be a significant global issue facing us over the coming years. So what do we know drives and causes these disparities? So there's several buckets of factors that are known to contribute. So environmental factors are among some of the most quoted. For example, our exposures to environmental toxins, air quality, transportation, where we live. The places where we live contribute to our disease risk. That instinctively makes sense to us. Behavioral factors such as our diet, our level of exercise, our adherence to health behaviors that contribute to our overall health. Social factors including health literacy, the level of education, our income, as well as our ability to access health care. And then individual cultures. We see differences in incidence and mortality in different places. All those places have different localized cultures and different beliefs that contribute to their health outcomes. Psychological factors like stress have been reported to be associated with cancer incidence and outcomes. And certainly for many of us in the room who've come to this problem, being interested in biological and genetic determinants of disparities in cancer. When you see these factors listed like this, it's difficult to think of a framework to put it all together with. So the AACR has this organizing framework they recently published in their Cancer Disparities report that looks at how these factors interact together in a framework that is driven upstream by structural factors in the US, such as racism, which lead to structural inequities and societal injustices, which acting through all these factors jointly contributes to cancer inequities and disparities across the cancer continuum. So when we refer to the cancer continuum as, risk, early detection, cancer development, treatment outcomes, and survivorship. My particular area of focus and interest is in the biological and genetic factors. And so we've come at this from this sort of epi lens of racial and ethnic disparities in cancer, but I've also told you that I'm interested in the biologic and genetic drivers. So there's a little bit of incongruence here because I'm interested in a biologic and genetic aspect of disparities, but it's defined by a social construct. So I'm going to take a little bit of a segue here to talk about the social constructs of race and ethnicity. So race is defined by as a category of humankind that shares distinct physical traits, or ethnicity by how groups identify according to common sort of racial, national, tribal, or religious background. So it's how people self-identify based on where they come from, what they believe about themselves. It's not an inherently biological variable. In the US, these are typically taken from standardized variables from, say, from the US government OMB, Office of Management and Budget, from the census, where we tend to get a lot of these data from the health systems, or ethnicity, where we have two categories in the US, Hispanic or Latino, or not Hispanic or Latino. So in thinking about disparities, many of us have started to consider that we want to understand how people are related to each other biologically. Perhaps a better lens to look at this through is one of genetic ancestry. So we can take the genetic data from individuals in our studies and infer genetic ancestry based on reference population, and look at how similar those individuals we're studying are to reference individuals in a reference database, and use this reference genetic ancestry or genetic similarity as the basis of grouping people together, and then determining how that relates to various cancer phenotypes. And so my group particularly studies ancestry effects in cancer and uses that as a foundational framework and organizing principle to look at how the social, environmental, and lifestyle exposure interact with our underlying ancestry-related genetics and biology, and manifest as cancer disparities in the phenotypes. The basis of this work, which I don't have time to go through this afternoon, is some of our earlier work, some of work I did as a postdoc with people, some of the investigators who are here at the Broad, is when here in the TCGA Atlas, in the pan-cancer Atlas phase in 2018, 19, thereabouts, we did exactly this. We inferred genetic ancestry from the germline data in the TCGA cohort, and then we looked at how that ancestry correlated with somatic phenotypes in cancer. So here, for example, this is work led by Jiang Carol Zhang, who's now at MSKCC, where she showed here, for example, that somatic alteration frequencies in particular genes in pan-cancer atlas, we saw elevated mutations, somatic alteration frequencies in these three genes, and then specific alteration frequencies in kidney cancer and VHL, PRBM1 and bladder cancer, and a couple of other genes specific to East Asian ancestry. So there is a basis of seeing molecular traits, so somatic alterations at various levels of biological organization, whether it's somatic mutations, expression, gene expression alterations that are related to ancestry in cancer, that could play a role in manifesting in our investigations of ancestry-related traits driving cancer phenotypes in disparities-related work. What is confusing is that in this work, both concepts of race and ancestry play a role and feed into the health status. And I found this schematic by Tim Rebick to be particularly insightful, because both our racial and ethnic identity and our ancestry have distinct and interrelated impacts on health status. So in essence, the underlying ancestry relates to biological attributes that contribute to the genomic architecture of an individual at birth, and those biological attributes that are influenced by inherited genetics and the lifetime exposures elicited on our epigenome jointly contributes to our health status, right? But our lifetime exposure and our experiences over the course of our lifetime, which contribute to us developing cancer, are also impacted by our race and ethnicity, because they influence where we live, what we eat, who we interact with, and our lived experience of the world that's translated into our biology via, say, for example, epigenetic mechanisms. So it's actually quite a gnarly ball of yarn to uncouple and twist, really, to tease apart the relative impact of ancestry, race, ethnicity in driving different cancer phenotypes that can contribute to disparities. And so the organizing principle of my lab is to analyze, as a computational biologist, is to analyze molecular profiling data sets, multi-omic profiling data sets from cancer cohorts to try and infer prognostic markers or predict clinically useful endpoints through a race, ethnicity, and ancestry, and determine sort of the biological and genetic factors that contributes to cancer health disparities. So this is our current mission statement as a lab. We ask questions like, what are the genetic and molecular features that drive cancer disparities? Does this lead us to a mechanistic understanding of cancer disparities? And can we build better models for prediction? And ultimately, can we identify novel therapeutic targets? That's the end goal. I'm going to give an example of a cancer type we work on today in the interest of time. Endometrial cancer is one of the tumor types we're focused on. It's particularly interesting to me, and I'm going to give a call to action for those of us who are here. And if you're online, please come work on endometrial cancer, everybody. It is a rising global epidemic, which somehow no one seems to have heard about. Incidence and mortality of endometrial cancer, uterine cancers broadly, is rising across the board. So we have incidence data here and mortality data on the right. You can see that the prevalence seems to be higher in higher income countries, although we see, for example, here that mortality is also generally high in low income countries. But the rate data shows a steeper increase in low-income countries. I'm not showing that data here. Early work from people who are here, including Gady Getz and Andy Cherniak, established that we have pathological classification, so classic histology, like endometrioid subtypes or non-endometrioid subtype by histology, and tumor grading associated with particular somatic alterations like TP53 mutations, and establish a molecular classification taxonomy as well, which lately has been integrated into clinical treatment guidelines as of a recent release of the FIGO classification in this disease. But what's particularly concerning, whereas other cancer types, the incidence and mortality is decreasing slowly over time, so we're making progress. For some reason, in endometrial cancer, it's the opposite. Even when we adjust our epidemiological estimates by rates of hysterectomy in this disease, we're seeing that the incidence rates for endometrial cancer are on the rise, and not unsurprisingly, the incidence and mortality rates are increasing at an even steeper pace in women who self-identify as Black. So Black women have the steepest increase in incidence and mortality, and they tend to get the most aggressive molecular subtypes, molecular histological subtypes that have less treatment options that we don't understand the genesis of. Okay. So one of our research questions is what's driving this disparity in aggressive disease presentation. And like many examples, we lack the cohorts that are racially and ethnically diverse, that have data that's collected across all these domains that we know are drivers of disparities. So we know what the problem is, we know what kind of data we need, and we know who we need it from, but we don't have it. Okay, so that's where we are today. And so I present to you what we're currently doing and where we'd like to go. So we got to start somewhere. We can't just sit around and say we don't have anything, so we can't do anything. So the first thing we're trying is to leverage existing clinical sequencing data we have. I work at a hospital, we see patients, and so we're looking at retrospective cases. We're in a large metropolitan area of New York, Greater New York Metro, one of the most diverse areas in the country. And so here we have a cohort over the defined area, the last 10 years. It's around of people who were seen in our hospital, who met the inclusion criteria, who also got clinical genome sequencing done. In that cohort, we're seeing around 30% African ancestry individuals. We look at them by histological subtype, we look at them through ancestry, and we try to correlate these and try to understand if we see an enrichment of specific mutations or characteristics of their tumors with ancestry, et cetera. And then we're also applying, because we're part of the hospital, for access to their historical residential addresses to be able to do geocoding and then use population or census data to get access, say, to their income and use indices of their socioeconomic status, population level indices, to estimate sort of their area level metrics of access or socioeconomic status. But we know from studies that were shown, so for example, this was a New England Journal of Medicine article that showed, for example, that one of our most popular clinical genome sequencing companies, over 80% of the people that have tested to this point, which was September 2022, were of European ancestry. So we can do what we can do with what we have, but it's not going to be sufficient. We are also leveraging this institutional catchment area we're in. We're partnering with other local New York area hospitals to build resources like this New York Polyethnic 1000 cohort, where the goal is to intentionally recruit historically excluded populations into these cancer genome sequencing cohorts, of which one of them is for endometrial cancer. The pilot data is here, where we show that the vast majority of individuals that were recruited in the study were of African ancestry, and then a subset of this cohort is endometrial cancer. We're currently analyzing that data now, not authorized to talk about it exactly and show you, but we're making significant progress and hope to have that data out later this year. But the cohort size is on the order of 100 or so. We can also do something with what's available right now. And so we're also trying to take advantage of population biobanks that are available, right? So we have All of Us. It's one of those resources that actually has genetic, socioeconomic, environmental, clinical, all these different drivers of disparity type data that's there. And so we've started working industriously, I would say, my team is, on defining case control cohorts in All of Us, so people with cancer, with endometrial cancer. Admittedly, the case count is low, depending on how strict you are with your case definition, anywhere from the order of 400 to 1,000. But if you consider that the TCGA cohorts, our own institutional cohorts, are in that realm, this almost doubles the number of germline endometrial cancers that are in the public domain. And then here we're defining concepts that are both genetic and social determinants data. We're using the survey data as well as clinically abstracted data from the EMR and building integrated machine learning models to predict endometrial cancer status. Ultimately, we want to predict risk. We want to build risk models, but it's still early days for us here, I would say. These three paths, I would say, are an imperfect solution because these data were not collected fit for purpose. And so I think what's on the horizon for us that's the most exciting is being part of the recently funded Cancer Grand Challenge team, the Cancer Inequities team, titled Team SAMBI, Societal Ancestry and Molecular Biology Analyses of Inequities. Very excited about this work. This work is being led by Dr. Melissa Davis at Morehouse School of Medicine. Basically, from everything I've told you so far, it's centering the African ancestry, African diaspora, and then looking through putting that at the center of the lens and looking at all these aspects that we know drive disparities in a fit-for-purpose way. The planned recruitment for this project is the US, the UK, and sites throughout Africa. Germline, tumor, normal match sequencing, RNA-seq, long-read, any sort of state-of-the-art technology in the right population with the right study design, with all the right known risk factors straight from the jump. But this is a 5-year project. So We're 3 months in? So very exciting, but this just goes to show that in solving and tackling a complex problem like this, we have to start from somewhere. So we start with where we are, but we know where we need to go, and projects like this are where things really need to go. Intentional study design, intentional recruitment in the right target populations. And so in essence, we want to focus on teasing apart the biological and genetic determinants of disparities, asking the right questions, and that's it in a nutshell. I'm looking forward to our conversation. We cannot do this work alone. I want to acknowledge my group who are working very hard. We have an army of collaborators that we're working with, all the sources of funding support. And today I especially want to acknowledge somebody very important to me, my postdoctoral mentor who passed away earlier this year. It's particularly impacting me this week because the Memorial Symposium celebrating his life's work and his contribution to science is this weekend on Saturday. And I put that link in there. There will be the symposium will be streamed live as well. It's on Saturday. So thank you for your attention.
All right, I've been looking forward to having your challenge, please.
Thank you, Nyasha, for your presentation. We're now going to move on to a fireside chat portion with Dr. Alicia Martin, our very own. She's an assistant investigator at the Analytic and Translational Genetics Unit at MGH, an assistant professor at Harvard Medical School, and an associate member at the Broad Institute, affiliated with the Stanley Center for Psychiatric Research and the Medical and Population Genetics Program. As a population of statistical geneticist, her research examines the role of human history in shaping global genetic and phenotypic diversity. Given vast Eurocentric study biases, she investigates the generalizability of knowledge gained from large-scale genetic studies across globally diverse populations. She's particularly focused on ensuring that the translation of genetic technologies via polygenic risk does not exacerbate health disparities induced by those study biases. Towards this end, she's developing statistical methods, community resources for genomics, and research capacity for multi-ancestry studies, particularly in underrepresented populations. Take it away. Thank you.
Thank you for giving the opportunity to have a conversation with such an esteemed, lovely colleague here. And thank you so much for all of your work on these disparities. I think they're so incredibly challenging to disentangle, so hard to understand. I want to start also with my condolences for the loss of your mentor. I'm sure that that is a huge loss in the field. I want to kind of start big picture in thinking about these disparities and having this chat with you, because you're interested in the biological, you're interested in the genetics, you're interested in the mechanism, but it's really hard to start at that level when there's so much ensconced in our society and in how, structural racism is impacting disparities, including through access to healthcare. So first I want to start with how do the disparities that you're describing and that you're researching relate to who can get care, how easily can they get care, and also related to trust in mainstream medicine?
All right, thank you. Before I answer the question, I also want to thank you for your work in this field. I think in getting to this field and tackling some of this work, reading some of your early work on GWAS and representation there was also insightful in us thinking about what we think about and getting to where we are as well. I think fundamentally speaking, I think that slide from the NEGM perspective was very telling, right? So we've done all this work in the genomics field to figure out the cancer drivers. So we're going to find all the drivers and find what's mutated and then find a targeted treatment and administer precision oncology. But we have this fundamental issue where some people aren't even getting the test, okay? And so even if there is a drug, they can't get it. I trained on the side of the analysis and identifying the mutations and getting to what drugs we need to make against which targets. And I didn't fully consider the ramifications of what if somebody didn't get the test. And so now that we're here, I'm having to really take the time to educate myself about these issues and consider the wider ranging impacts of that. And I think it comes back to a question and discussion we were having earlier here today with colleagues, that I don't feel like I'm an expert in access issues and, et cetera. But as a practicing scientist, I have to be informed and I have to be aware of the science we're doing has an impact in society. And so being more mindful about communicating what we do outside of my realm, outside of my genomics colleagues, talking to people. I think at the recent AACR Disparities meeting, it became clear that a platform for patient advocates, and advocacy in general for scientists is important, because that message needs to go out to these communities so that people understand what it is we're doing, what genetic testing means, what it means, so that it becomes part of everyday language, part of the everyday lexicon, so that if your physician asks you to be part of a study, it's not something. I believe a patient advocate called it. You're not a guinea pig in a study, right? To be part of a clinical trial, so there is some responsibility on us as scientists to advocate a little bit and to communicate our science more widely, so that what we do has a wider impact and influences legislation, which in turn will lead to changes in who has access, what gets paid for, and then who is represented ultimately.
Absolutely. I feel like it's a cycle that we all need to kind of play a role in, especially as scientists understanding the data. On the communication angle, science communication to marginalized groups is very important. So on that topic, beyond the analytical work that your group is doing, you mentioned a vested interest in the science communication piece. What is your lab doing, and beyond the scope of the analyses, to try to sort of bring everybody along and make sure that people don't feel like guinea pigs when it comes to such a massive kind of life or death kind of decision as what kind of treatment you get?
Yeah, admittedly, I don't know that we have formal things we're doing as a lab, and as the lab head, that falls directly on me. I am a shy person, believe it or not. So I'm like, as soon as these lights go off, I'd like to run out that door. It's hard, and I've been thinking of getting maybe more formal training, or actually, seeking out maybe more formal training and building even just a 5-minute spiel or 1-minute spiel that I can use to communicate these things. I would like to be better at social media. I've reached out to our social media department, and every institution has resources, right? Where our communications team have professionals. We are professionals at this. We have professionals at that, who can actually help you. You can meet with them, explain what you do, and they can help you create packages. I have a younger sister who is much more facile at technology than I am, and she schedules her social media posts and stuff. And I was like, looking over her shoulder, like, could you help me do that? Because the other thing I'll say is, I don't have time. And then she's like, well, you could schedule the posts. I was like, is that a thing? Okay. So I think, bringing more intentionality to it is definitely something.
To that point, I mean, also to your credit, this is something that you want to do, but as with many folks in this room, I'm sure we don't have time for all of the things that we would necessarily like to do. And there are communications experts and their advocacy groups, and playing a role at liaising between these communication experts and advocacy groups seems like something we might consider as scientists who deeply understand the data and deeply understand some of these bases of disparities. Is that something you might consider in the future?
For sure.
Awesome. I wanted to come back to this idea of the environment. So it's, such a big topic.
Right.
We're all very curious and excited about the environment. And I'm coming sort of from like a bit of a GWAS perspective where like we're doing massive genomic studies all the time. But to the points on some of your slides, some of these types of omics data don't require hundreds of thousands of people to be particularly meaningful to patients. So I'm curious about some of the like cancer risk projects that you're contemplating and that you're working on, and particularly in low and middle income countries, where scaling multi-omic data may be a particular challenge. What are you thinking about the utility of some of these omics data to play a role in both understanding the like molecular profiling nature of cancer, but also to potentially bridge a gap with our understanding of the environment and social determinants of health, and maybe trying to build some causal models that we can use as a framework to try to understand how these different pieces all kind of fit together?
Excuse me.
I'll give you a sec to think about that and just get some water. But for example, we're helping build biomarkers from proteomic data on the basis of smoking, which is obviously a big risk factor for lung cancer, for example. Are you thinking about these kinds of things? And where are you excited about the field going on the cancer disparities topic?
Sorry. Yes.
You're good.
No, this is an exciting, very exciting set of points to bring out, especially on the issue of scale, right? Because we can't all be part of these large consortia that have $1 billion to do 20,000 samples, right? And I think this goes back a little bit to what I was saying about practicality. Like, what can we do today? We all have a collaborator who has some number of samples. And so the question is, what can we do with what we have right now? And how can that help? And I really like this example you gave about smoking, right? I think the other omes, like expression, proteome, metabolome, potentially have informative information there that really gets at biomarkers, really gets at doing something, getting to a place where you could design a low-cost test or screening diagnostic very quickly. And so things like that are very exciting. I did my PhD in DNA methylation-based subtyping in cancer, and DNA methylation will always be my first love. And I've been doing a lot of thinking lately about, reading a lot too about the epigenome as a mechanism of embedding of environmental exposures, and thinking a lot too about genetic regulation of DNA methylation. And so triangulating across those, there's some thought been given by other people of building methylation risk scores as indicators of the general environmental exposure signature as well. So sort of a fingerprint of where you've been and how it's been going. So there's lots of little areas like that you could probably get started pretty quickly. One thing too I like about the epigenome is that you're using DNA, and with our logistics and fixation, that has the potential to stay a bit more stable over time and across conditions, which might be a challenge in some type of environment. So there is some room there for exciting developments.
Definitely. And one of the areas that you were talking about was in endometrial cancer, this sort of explosion in prevalence that we might expect during the next few decades. And I'm wondering if there's a good understanding of what the exposures are that are potentially going to lead to that explosion, and whether there are some omics data that might help tag those, because that's an interesting and exciting area, I think.
The biggest risk factor that the biggest driver of that rise is obesity and other metabolic syndrome. And so just like in general in the world, everything related or, triangulating with the obesity epidemic in the world is converging there. And then there is hormonal exposures as well, of two big things that are also potentially easy to measure. There is potential to intervene, but I think it feels wrong to sit here and say, oh yeah, obesity is easy to tackle. Because if it was, we would have gotten there already.
Yeah, similarly on the hormonal side, I'm sure there are some hormone blockers, but I'm sure also that impacts a whole system, not just a single component of your risk.
I think one of the challenges in endometrial cancer is that it's relatively understudied, right? And so what we thought, for example, there was like type 1, type 2, this one is hormonal driven, this one is not. It turns out that it's not quite that simplified or that one or the other, and of course, it's always a continuum. And so just having more directed attention and more rigorous study will be important.
Awesome. Well, I'm looking forward to seeing your role in that. Kind of a general question, again, zooming out, that we were kind of discussing is, where do you start with your research? You're exploring such a massive space, and what determines which questions you want to be really going after? For example, some genes have known cancer drivers associated with them. Do you start by looking at those drivers and in those specific cancers, or do you take kind of an unbiased approach? And if so, how? Maybe that's a tough question.
It is a pretty tough question. It depends. It depends on the project. It depends on the focus. And so when I started my lab, I think it also depends where you are in your career, right? So some projects are longer term investment, right? So you start, you want to take an unbiased approach and you plan the study well, and it takes how long it takes, right? And some projects are student driven and they have to graduate in 2 years. So you design a project where you've got a fair idea of where it's going, and it's an interim objective to the longer term objective. So you're more focused. You have a specific hypothesis about a particular pathway, and that project is constrained to that pathway. So then that's what determines whether you focus on specific genes or specific pathways. I'm looking at the presenter screen. It's still showing my mentor, and one thing he always used to say was, let the science lead you. So I think I should stop talking.
That sounds like wise advice for all of us.
Yeah.
I want to turn to sort of a question that's kind of near and dear to my research and our shared interests, which is the topic of ancestry. So you, I think, gave like a really nice outline of the delineations between race, ethnicity, and ancestry. So I just wanted to start with, when researchers are studying cancer disparities, whether that be in incidence or mortality, do they use, just to reiterate, ancestry or self-identified race or ethnicity?
I would say they use self-identified race or ethnicity, particularly if it's epidemiological data, because of how that data was collected historically.
Mm-hmm.
So, for example, the surveillance database that's maintained by the National Cancer Institute, the surveillance database that maintains that data, collects that data using self-identified race or ethnicity.
Yeah, and I guess that's something that you can easily get from a cancer patient walking in the door without a whole genome sequence or without genetic data already generated. So when you're doing cancer genomic analysis, are you using ancestry, or using race, or using ethnicity, or some combination of these?
I would say looking at it from an ancestry perspective is fairly new. I think, I'm trying to remember when I first started seeing papers looking at it from that perspective, but I think for a long time in cancer genomics, we looked at the cohort as, this cohort as this tumor type, in these mutations and didn't consider necessarily racial ethnicity. Yeah. And then for a long time then, when people who studied disparities would compare mutations, they would compare the rate of mutations between groups defined by self-identified race or ethnicity. And then people started asking, what is the rationale for expecting differences from groups that are defined by self-identification? So then it's like, oh, right. If it's a social construct, can you expect that you'd see differences in mutation? Then if you see differences in mutations by those groups that are defined by socially defined variables, is it the social definition or what's genetically similar between those two groups? So that's, I think, how sort of it converged to, oh, maybe we should define the groups by genetic ancestry. Then if we see differences, then we can know that these differences are genetically driven.
Right. I guess you have like a bit of a chicken and egg kind of problem when you're trying to understand and tease apart these disparities. Because if you're doing a germline genetic study, differences in allele frequency that you're identifying, you want to make sure that you're controlling for ancestry. However, if you're then looking at driver mutation differences, maybe that differs as a consequence of a whole lived experience, and maybe that's a little bit more social. So at some level, I see utility in having both of these when trying to understand and study disparities as they relate to the genome, whether it be germline or somatic in this context.
Oh, yeah. I don't know if we have the kind of time to delve into it, but I've been thinking about this, and maybe you're the right person to because in a GWAS, right, you add all your admixture coefficients, right, to control for the effects of ancestry, to find SNPs linked to your trait. But what if your trait is inherently ancestry? No, your trait is the disease, but you can take two approaches, right? You can do the GWAS in each group independently, but then you're losing power because you don't have that many samples from the non-European ancestry group.
Yeah, there's a lot to do. Yeah, that's a tricky one.
Right.
So for the GWAS, you want to make sure that you're accounting for ancestry to ensure that any allelic effect on the disease is larger in, say, cases compared to controls versus background expectation. But then when you're getting to what is the driver of some of these cancer differences, and you're bringing in all of these social and environmental contributions, structural racism, access to health care, obesity, hormonal regulation, or hormone exposures, there's so much there that can also influence, does someone get cancer in the first place? So bringing these two things together, both of them are impacting the genome in some way.
Right. So I've seen some strategies people started to employ, for example, but it's also circular. So let me put it this way. You can say, for example, use a PRS to control for the genetic effect, right? If you have a PRS that works well across all groups defined by ancestry, you can add that to your model to ask, given that you have this genetic background, do these social effects play a role in contributing to the disease?
That's makes a lot of sense.
One approach. In endometrial cancer, there's been one very big GWAS. I'm unsure. Where's the camera? I am unsure whether that PRS works equally well across all groups.
Well, I think you and I could talk about this topic all day, but to broaden the conversation so other folks in the room I know will be also really interested, I want to also talk a little bit about the All of Us work that you're doing, particularly because that's such an amazing biobank that's US-wide, and it also is intended to boost representation from traditionally underrepresented groups in biomedical research. So how much information is encoded in All of Us that is useful for us to dig into trying to understand some of the root causes of cancer disparities?
I think as designed, All of Us is one of those unique resources that is optimally designed to help us to tease apart some of the questions we have about disparities in general, about diseases in general. For cancer, it works well. It has one small drawback in that we don't have tumor genome sequencing, but we do have germline sequencing data there. And then we have all the social and some environmental data, and then we have all the clinical data that you can extract from EMRs.
I wonder if there are going to be future researchers that are like, this is a gap we need to fill. We're going to go sequence those tumors too.
Right. I'm not sure in the roadmap if the tumor samples were collected, but if that was there, then it would be a gold mine.
Yeah,
I would certainly sign up for access.
Definitely. What else in the All of Us sort of data would be amazing to have? I know, for example, from working with biobanks that cancer registry information can be a little limited, including with things like staging and histology and that sort of thing.
Yeah, definitely. I think for cancer utility, having more of the pathology type information would be ideal. So having more pathology confirmed, histology, staging, and then an easier way to get survival and update on survival outcomes would be like icing on the cake.
Nice. So good to have, but you've got a starting point already.
Right.
Great. Kind of on that topic, since All of Us has whole genome sequencing data, what information can you learn about someone's likelihood of having cancer, what type of cancer they might have from their whole genome sequencing data, germline data alone? And then relatedly, what other pieces of information would be really valuable to have?
If you just had the whole genome data alone, you could not determine whether a person had cancer or not. But you could determine the risk of having cancer based on triangulating the whole genome data with databases with known pathogenic germline variants, especially those in highly penetrant germline cancer predisposition genes. So for example, again, I'm just going to use endometrial cancer today. I do know about other cancer types. We have cancer predisposition syndromes like the Lynch syndrome, which predisposes you to endometrial and colon cancer. So you have a defined set of genes that you know pathogenic variants in these genes leads to high likelihood of getting endometrial colon cancer. We can also, again, estimate the likelihood of developing that tumor type using a PRS, a polygenic risk score for different cancer types. We can, what else can we do? Oh, we can look at the structural variation data. That would be very important to look at sort of large scale duplication or structural alterations in the genome. Those would be very, very telling and predictive. But I think the ultimate power of All of Us comes from having multiple streams of different types of data to work with.
Awesome. This series is sort of co-run by the Equity and Biomedicine series, and also is being set up by Women at Broad. And so I want to turn a little bit to some career development question, because of course you're running a lab, you're working on cancer disparities, your career trajectory seems like one that's fairly inspirational. So can you tell us a little bit about the lab that you've built and what you look for in a team, and any sort of career advice that you can lend to folks in the room?
Right. So I've had my lab for 2 years, 8 months, 4 days, 3 hours. 2.5 years, give or take. And I love it. I really love it. I love my group. I think there are many things that one could do with their PhD. This is one of them. I love I love the work we do. It's hard sometimes. It's fun sometimes. It's a lot of things. And so I come from a background of team sports. So when I was building my team, I really thought about how, a lot about those principles of how everyone on the team, we're all working to one goal. We want to win. Whatever, how you define win, publish the papers, eradicate cancer, advance our careers individually and collectively. But not everyone is the same. Because if we were all the same, like let's take, for example, a basketball team. If we were all tall, then nobody could shoot. Or if we were all dribbling the ball up, then nobody could get the rebound. So it's really been so far, the first few people who've joined the team, they are not all the exact same phenotype. And so I think there's been strength in numbers in that way, in that we don't have a single person who can do, we don't have any single copy of the same person.
That makes a lot of sense. I guess building on that, what advice do you have for trainees who are going out and are thinking about what their next steps are, be that from a post-bac position or having done their PhD? What comes next? That was, at least personally, like a pretty existential kind of moment of self-reflection, and it was really challenging. And so I'm wondering how you came to your position, how challenging it was to get to your position, and do you have advice for those that might be thinking about what their next steps are?
Indeed, it was. I identify with that as well. It was extremely challenging, and it took a pandemic to bring the level of self-examination that determined my course. So the first piece of advice I always give is to spend a significant amount of time in self-reflection to decide what makes sense for you. There's a lot of advice that's going to be handed out to you about what makes sense to other people, what they would do, what feels good for them, what's right for their life, which might not be right for your life. The second thing is to talk to people. So having the right team of mentors, not one person. You could have a primary mentor, a secondary mentor, but talk to as many people as possible. You can have one question that you ask to everyone that you talk to, and different questions you ask to different people to try and get sort of a 360-degree perspective on the issue. And then I think there's some level of serendipity to what happens as well, where you are, and then there is just some encouragement for somebody who's out there and who's in this place. Because there was a time where I would say I was really like, I don't know where I'm going. I don't know where this is going to be. And now, I guess 3 years down the road, I'm sitting on the stage. I'm apparently giving an expert talk. I got a grant from the NIH. And so if you had asked me these questions 3 years ago, I might not have been as perky about it, because I was in a place where I thought, I'm not sure this next step is exactly where this is going. So I just want to encourage other people that at that moment, it might not feel like things are gonna turn out, but just keep persevering. You might just be in that last dark space just before things turn bright. And so I just want to, encourage somebody out there today.
Well, great. On the optimistic note of perseverance, I think we're at time.
Exactly.
Great. So thank you so much for such an illuminating, wonderful conversation. It's been such a wonderful opportunity to meet you. I really appreciate it.